Welcome back to the playlist on transcription. So what we did in the past few videos is we talked about transcription and how transcription is basically a process by which we use the DNA inside the nucleus and we synthesize something called an RNA strand. And we mentioned that there were several types of RNA strands. For example, in this video we're going to be talking only generally about mRNA, but there are other types of RNA strands like transfer RNA, ribosomal RNA. There's also some stuff called small nuclear RNA. So there's lots of different RNA strands that we can use around the cell. And transcription is a process by which we synthesize all of those. But when we're talking about mRNA, mRNA has to be processed a little bit before it's able to exit the nucleus. Okay? So here's the idea behind mRNA processing. Okay, we're going to have several steps, and I'm going to outline them for you now. You have an essay on this, so what I'm about to do is outline the steps of mRNA processing. So the first step, I'll call this step 1. So the first step of mRNA processing is something called 5' capping. And essentially what we're going to add is something called a 7-methylguanosine cap. Okay, this is a protective measure that the cell uses to protect the mRNA from hydrolysis. And as we'll find, it also serves as a recognition part for the ribosome. Okay, we won't talk very much about that. The next step of what's going to happen is we're going to splice out these things called introns. These are parts of the RNA that don't code for amino acids. So we splice out the introns and effectively glue the parts that do code for amino acids together. Okay? Then we're going to use something called an endonuclease. So intron splicing, we could call that essentially step 2. Then step 3, we're going to use an enzyme called an endonuclease, an mRNA endonuclease. And we're going to cleave off a segment of the mRNA. And we'll talk about that in a few minutes. And then the last step is something called 3' polyadenylation. So these are essentially the 4 steps that we're looking at in terms of mRNA processing, and we'll go through these pretty quickly. What I want to show you essentially is what we start with after we have transcription. So in a lot of cases what will happen is this base right here, let me scratch this out. This base will a lot of times be an adenine. Okay, so what we essentially have is we have this adenine base with 3 phosphates right here. So we have these 3 phosphates right here. Okay, if you look at the abbreviation, these abbreviations are what I essentially want you to know. Okay, so what I have here is, okay, here's a phosphate. This is a phosphate, and then I have some nucleotide, and then attached to the nucleotide I have this triphosphate right here. Okay, so whenever you're telling me the abbreviations for each one of these groups, what you'll start with is this nucleotide, where the rest over here, all of this is the rest of the mRNA. You'll have a nucleotide, and then attached to it on the 5' end, you'll have 3 phosphates, okay, in a covalent linkage. So these right here, this is the triphosphate linkage, okay? Now, if you're in the A&P, you can sort of take a step back right now. I'm going to explain something for the people who are in biochemistry, okay, and it's the following. So if you look at this structure right here, you notice we start with a nucleotide triphosphate on the 5' end. Okay, this is the 5' end. And you go back and you take a look at RNA polymerase's mechanism, which, by the way, again, is not on your A&P exam. This is for the purpose of biochemistry. You'll notice what essentially happened. You'll notice that the rest of this going up here, this is, of course, the remainder of the mRNA strand that has already been synthesized. And what we said is that you had 3' hydroxyl group nucleophilic attack on the alpha phosphate, which is shown right here, of the incoming nucleotide triphosphate. So this right here is the nucleotide triphosphate. Here's the nitrogenous base. Here's the ribose, and then you have the beta phosphate here and the gamma phosphate right there. So the 3' hydroxyl group of the already synthesized mRNA strand does a nucleophilic attack on the alpha phosphate of the incoming nucleotide triphosphate. And of course, what ends up happening is all this business over here, this is pyrophosphate. This is the pyrophosphate right here that leaves. And so what you get is essentially the 3' hydroxyl group here is now attached to the 5' phosphate of the next nucleotide that we added. You'll notice that for this to happen, for you to get nucleophilic attack on the alpha phosphate of the incoming nucleotide triphosphate, there already has to be a pre-existing nucleotide up here. But if you go to the structure that we're talking about for the 5' end, there wasn't. This is the first nucleotide that was added. So this is the first nucleotide that was added. So there wasn't anything before it to do a nucleophilic phosphoryl substitution on this phosphate right there. Okay, so that means that we never lost this pyrophosphate right here. This pyrophosphate was never lost. So that means we still have the triphosphate there. And so what we're essentially going to do is we're going to add a 7-methylguanosine cap to prevent hydrolysis of these phosphates. Okay, so let's talk about how that occurs. So the first thing that I want you to notice is these abbreviations right here. The lowercase p's are for phosphates, and now this is where I want to bring it back to A and P. These lowercase p's are for phosphates. The N is for some generic nucleotide. It could be any one of the 4 nucleotides that we have in RNA. It could be an adenosine, a guanosine, a cytosine, or a uridine. It could be any of those. And then we have these 3 phosphates on the 5' end. Okay. What I'm going to do now is I'm going to use an enzyme called phosphohydrolase. Okay. What this enzyme is going to do is it's going to split the bond between the beta and the gamma phosphate. So notice what the product of this reaction is. Okay, you still have this nucleotide right here, right? Of course, you have the rest of the mRNA strand going towards the 3' direction. But now, because I hydrolyzed off a phosphate, notice there's only 2 of them left. And that's what gives this enzyme the name phosphohydrolase. It's hydrolyzing off a phosphate, and so you end up now with only a diphosphate linkage. If we want to look at the structure from a biochemical perspective, what you should see is that now there's only 2 phosphates attached. Okay, that's important for the next step, and the next step is essentially called guanylyl transferase. So the substrate for this particular reaction, which you do need to know the substrate is called GTP. GTP is just another ribonucleotide, triphosphate, that we have floating around the nucleus at this point. And essentially what's going to happen is a guanosine monophosphate. So the guanosine monophosphate part of GTP is this right here. This is the guanosine monophosphate. This guanosine and phosphate are going to get transferred onto the diphosphate. So what we have here is we now have attached the GMP. So this right here, this is guanosine monophosphate. We now attached that to these 2 phosphates right here. And then, of course, I still have, let me do this in green, I still have the nucleotide right here, and the rest of this extends for the rest of the mRNA chain towards the 3' end. Okay, so I've attached a GMP onto this. So this whole business right here, okay, these were the 2 phosphates. These were the 2 phosphates right here left over from the previous reaction of phosphohydrolase. And when I use the guanylyl transferase, notice what happens is I essentially transfer a guanosine moiety. And that's what gives this enzyme the name guanylyl transferase. I just transferred a guanosine monophosphate. So this whole business up here that I circled in red, this is a guanosine monophosphate. And the substrate for this reaction, notice, is GTP, and I'll get out pyrophosphate. Okay? Now, in terms of the next reaction, what's going to happen is I'm going to methylate the nitrogen on the guanine ring. Okay, so if I was to circle the guanine ring, it would essentially be all this business up here. So this business in here, this in this little segment, this is guanine. Okay, so the next enzyme is called guanine 7-methyltransferase. So notice what happens in the next picture. Okay, this is the methyl group that I just added, and notice it was not present here in the nitrogen of guanine. So what I essentially do using a molecule which is abbreviated as ADOMET, this is also called S-adenosylmethionine. You'll see it abbreviated as SAM. In fact, on the exam, if I ask this, I probably won't ask this essay, you're welcome to call it SAM. And SAM transfers its methyl group to the guanine. In the process, you get out S-adenosylhomocysteine, or SAH. Adohomocysteine is the same as S-adenosylhomocysteine. So SAM, in other words, in this reaction, this is the methyl group donor. That's the methyl group donor. And notice where that methyl group ends up. It ends up being attached to this nitrogen right here, and thus you see the methyl group right there. Okay, hopefully that makes sense. The last enzyme that essentially acts is also an S-adenosylmethionine dependent enzyme. Okay, so notice this one also requires adenosylmethionine, or ADOMET. So this is also an S-adenosylmethionine dependent enzyme, and ADOMET, or SAM, is going to transfer its methyl group to a different position. Okay, notice that in these molecules, right, let me circle this in yellow. Actually, let me do this I'll do it in green. So this moiety right here, this is essentially the ribose ring, or what's left over from it when we attach it to things. So this right here, again, this is the ribose ring. Notice on the ribose ring I have a 2' hydroxyl group. So let me highlight that. Right here I have a 2' hydroxyl group, right? And in that hydroxyl group, the oxygen is attached to a hydrogen. So what essentially this 2' O-methyltransferase does is it's going to methylate the 2' hydroxyl group. So notice this hydrogen effectively, and others down here as well, this one as well, so let me highlight that. These 2' hydroxyl group hydrogens get replaced with, what is it over here? They get replaced with methyl groups. And again, this is catalyzed by 2' O-methyltransferase. And again, it is a SAM-dependent enzyme, S-adenosylmethionine. And in the process, you end up getting out S-adenosylhomocysteine. So when you're naming this stuff, okay, notice after guanine 7-methyltransferase, what you're welcome to call this is notice, I still have the 3 phosphates here. So here's 2 of them. These are the 2 phosphates right there. I still have this nucleotide right there. And then I have the GMP. So I have the GMP, and then you're welcome to put this M7 to denote that the 7 position on the guanine, the 7 position right here, let me highlight it, the 7 position on the guanine, which is this nitrogen, gets methylated. Okay, so this M7 is denoting that the guanine is methylated at the 7 position. Okay, also after the 2' O-methyltransferase, we have essentially the same thing. We have this 7-methyl guanine, which is this GMP right here, and then I also have these 2 phosphates, right? Still have these 2 phosphates, but now the nucleotide right here is now methylated. So I put this M in front of the nucleotide to denote that it's methylated. And of course, I still have the rest of this mRNA over here towards the 3' end. So what do you call this business in general? So let me highlight all this. All of this right here, all of this that I'm highlighting, this has a name. And actually, I name it right here, but let me write it so you can see it. This is the 5' 7-methyl guanosine cap. And this has several important functions that we'll probably elucidate in later videos. But suffice to say that in eukaryotes, like mammals, this process has to occur before the mRNA can exit the nucleus. Okay? Now, there are some other reactions that also take place, and these are a lot easier to understand, I think. Okay? So let's first define introns and exons. Okay? So what I've essentially done is I just ended up methylating I just basically ended up methylating the guanine, methylating some 2' hydroxyl groups, and I effectively made a 7-methylguanosine cap on the 5' end. Okay, so out of all of that RNA, that of course, all the RNA continues down over here, right, and on down to the 3' end. Well, out of that whole mRNA strand, okay, only some of it codes for amino acids. Okay, the component that codes for amino acids for the protein, that component is called the exon. Okay, an exon is a component of the RNA that codes for amino acids in the protein. There are other components of the mRNA that do not code for amino acids. They're just simply leftovers of the mRNA that don't code for anything. These guys are called introns, and these introns, we'll find, are going to get spliced out. Okay, these introns get spliced out, and so all you have left then are exons. Because if I take the introns out, all I'm left with are exons. Those are the only two things that are in the mRNA, introns and exons. And because the introns don't code for amino acids, it's advantageous to take them out, so by the time the mature mRNA gets to the ribosome doesn't really have to do any work, it just simply has to read the mature mRNA. So the introns will be spliced out, and all that's left are exons. All that's left are going to be exons. And so what you could effectively view it as is the exons are effectively glued together. So let me give you a hypothetical immature mRNA strand. So let's say that the red are the exons, and like I did before, the black ones are the introns. And just so you can see it, let's keep them separated like this. Okay? So essentially what's going to happen is, if this is, say, this is component 1, 2, 3, 4, 5, 6, 7, once I splice out the introns, right, if I splice out all the black components, what am I going to get out? Well, I'm going to get out 2, 4, and 6, right? So this 2 component, this 4 component, and the 6 component, all that stuff gets spliced out, and what am I left with? Well, I'm left with, once it's glued together, essentially just all of the exons, right? And that would be components 1, 3, 5, and 7. So what I've done in this process is I've spliced out the introns. So all that's left with and glued together are the exons. And those right here, these are the ones that code for amino acids for the protein's sake. Okay? And this process of splicing can be carried out in 2 ways. Number one, it can be carried out by catalytic RNA. We're not going to go into details of that. And then there's an enzyme complex that assembles called a spliceosome. And the spliceosome can also splice out the introns to glue the exons together. Okay? And kind of the way to think about it is the exons are basically sewn together with all the introns removed. So hopefully that makes a little bit of sense. Okay. So that's something completely different than what we're about to talk about. So let me kind of bold that off. So I want to point your attention to several things. Number one, this Pol II right here, what this essentially is referring to is RNA polymerase. Okay, that's what that's referring to. Now what's essentially going to happen is this enzyme complex right here, this enzyme complex is going to come in. Okay, and it's shown in blue. You can't really see it very well. In fact, let me sort of highlight what they're talking about. This enzyme complex is in blue. And so notice what happens is, while the polymerase is still attached to the DNA, this enzyme complex comes in here and binds as well. Okay, now once the mRNA is finished with its synthesis, once the RNA polymerase is finished synthesizing the mRNA strand, it dissociates. Okay, so notice here, RNA polymerase has dissociated. So this molecule right here, or this complex, is RNA. This is RNA polymerase. It dissociates. Okay? So what's going to happen now is we're going to take over with this enzyme complex. Okay? So the question is, what does the enzyme complex do? Well, the enzyme complex has a few enzymes associated with it. One of those enzymes is what I denoted as 3 right here. It's this endonuclease. The other one is an enzyme called polyadenylate polymerase. So the question is, what happens? Well, what I want you to notice is there's this sequence right here in the mRNA. Okay, it's this AAU-AAA. Okay, and yes, I would give you this on the exam. Okay, so I have this AAU-AAA. So there's AAU-AAA. So that's, in other words, that's adenine, adenine, uracil, adenine, adenine, adenine. Okay, this enzyme complex looks for that. Okay, it specifically looks for the sequence AAU-AAA. Okay, when it finds it, the endonuclease part of this enzyme complex clips everything after it off. Okay, so do you see this kind of tan part right here? Let me shade it in let's do green. So this whole part right here, all of that business, notice it's gone. Okay, look at the next picture. Do you see how it's gone? Okay, this component, this is essentially the 3' end of the immature RNA. Okay, it's gone. Okay, so notice how when you get to this picture right here, all that stuff has been removed, and all you have left is the 3' end. Okay, that is catalyzed by the endonuclease. And if you wanted to know what an endonuclease is, it's essentially an enzyme that finds some middle segment, somewhere in the middle of the RNA, okay, and it cleaves off everything to one side of it, okay? In other words, the place I'm targeting is essentially right here, right there where I'm highlighting. That's where the endonuclease is targeting, and all that business that I highlighted in green gets hydrolyzed off. That's essentially what an endonuclease does. And so it gets rid of all that business. So now all we have left is the 3' end of the part that says AAU-AAA. And notice that part's still there. Okay? And the enzyme complex that had endonuclease and polyadenylate polymerase targets that specifically. So hopefully that makes sense. Okay? Now what I'm going to do is I'm going to use polyadenylate polymerase. Okay? What this enzyme does is it uses the ATP molecules in the nucleus. So it uses a bunch of ATP molecules and attaches them sequentially to the AAU-AAA. So notice, I still have this AAU-AAA right here. But what polyadenylate polymerase does is it attaches a whole bunch of A's to the 3' end. So what you would literally see after this AAU-AAA is A, hopefully you get these on the exam, A, and off and off and off to a certain point. So that way, the 3' end is polyadenylated. Okay, adenylate, if you want to know what adenylate was, let me write that up here. Adenylate is simply a molecule of adenosine monophosphate, AMP. Okay, so literally what you're adding is adenosine monophosphates, but the A is just representative of the nitrogenous base adenine. So you can more or less assume that you're just adding a bunch of adenine-based nucleotides to the 3' end. Okay? Once that is completed, then the enzyme complex dissociates. I did that in blue. So this whole enzyme complex, I highlighted in blue, this whole enzyme complex dissociates. And now what you have at this point is you have the mature mRNA. So this right here deserves a bold color. In fact, it deserves all capital letters. This is the mature mRNA. Mature mRNA. Okay? And once we have this mature mRNA, it will exit the nucleus and head for a ribosome. But before we get to that point where we're going to look at the ribosome, I want to take a step back and kind of think about how this works. So this right here, okay, this is the gene for ovalbumin. This is not a protein this is not a gene for a protein that exists in humans, but here's the DNA for it. Now, what they've essentially done in the first step, so the first step, I'll call this step 0, that's transcription. Okay, that's the thing we described in the previous video. And then they did 5' capping. That was what I called step 1 in this video. Okay, so notice what happened is you do transcription and then you add this 7-methylguanosine cap, which is shown in red. The rest of this is essentially a conglomerate of introns and exons. So all this business in here is a conglomerate of introns and exons. Okay, the introns are shown with these letters. Okay, so anything that has a letter is going to be an intron, and the green things are the exons. So let me give you an example. This right here, this is an intron. Okay, something in green like this is going to be an exon. And remember, introns don't code for amino acids, but exons do. So then what's going to happen is the second step I told you about, which was splicing. The introns are going to be spliced out either through a spliceosome or through catalytic RNA. So those get spliced out. These introns are shown right here. These are the 7 introns, right? You can count them. There's 7 of them. And then the exons get glued together, right? The exons get glued together. That's part of the splicing mechanism. Then you have cleavage by the endonuclease. The endonuclease targets that AAU-AAA sequence and cleaves everything off from the 3' end of that sequence. And then the last step is polyadenylation by polyadenylate polymerase. And so you notice that on the last part here you have a bunch of these adenine nucleotide residues. And this right here, this is your mature mRNA. Okay, so hopefully that gave you a little bit of intuition on what happens to the mRNA once we transcribe it. We're not done at that point. We have to process it a little bit and get to mature mRNA. And actually, one thing that's interesting, I'm not going to do a video on it as of yet, but tRNA is also modified to a very great extent. We mentioned earlier on, and you're not responsible for this, but this you are. Basically, in RNA, we have several bases generally. We have adenine, uracil, guanine, and cytosine. These you do need to know. That's something you'll need to know for lecture and lab. But one of the interesting things about tRNA that you're not responsible for is tRNA actually has a great number of extra bases. And these are basically what we call post-transcriptional modifications. Okay, a post-transcriptional modification is where you take the RNA strand that was just made and you use various enzymes on it to change the bases into things that are not A, U, G, and C. So tRNA has what we call funny bases. You could certainly Google that and look at different structures, but I'm not going to do a video on it just yet. Eventually I probably will, but suffice it to say tRNA has some funny bases in it. Okay. But mRNA does not. mRNA is just simple A, U, G, and C. As far as I know, there's no exceptions to that. And these were the processes I just outlined to get a mature mRNA. So make sure if you're in my A&P lab, look at your essays. Use this video and the PowerPoints to find the answers to those essays. Hopefully I gave you a little bit of intuition on mRNA biosynthesis. In the next video, we're going to go to what we're going to do in week 2, which is talk about ribosome function. See you in the next video.